See you guys soon. Welcome, everybody. Welcome to InsideTheHive.tv, the show that takes you into the world of bees. Today, I have a special guest, Dr. Esmaeil Amiri from University of North Carolina in Greensboro, UNCG. Go Spartans. And today, we're going to talk about honeybee queens infection by viruses. Dr. Amiri is specializing himself. He has been publishing a lot in this subject. And I invite him to the show so we can discuss and know more about and educate ourselves about how honeybee queens get infected by viruses and maybe find out ways to avoid that. Let me bring Dr. Amiri with me. Hello, Dr. Esmaeil Amiri. How are you?
Hey, Humberto. How are you today?
I'm good. I'm so glad that you're here. We have so much to talk about. Before we got there, I just want to thank you, my Patreons, for the opportunity, for allowing me to do this and keep those videos and this live stream free to everybody. I'm very glad to have these people supporting us, and without them, I don't know what I could do. So yeah, a special thanks for the 18 Bees, DC Beekeepers Alliance, Union Bee Company for supporting us. If you want to know more about how you can support this channel on Patreon, visit patreon.com/InsideTheHive.tv. Thank you. Dr. Amiri, we have a lot to talk about. So let's jump into it. The way this is going to be going is, one second. Guys, please let me know if you can hear us well. If you can write something in the chat room, so I know everything is running well, so I can help to make a better experience to everybody. So, Dr. Amiri, the way I planned that, if you could give us a little overview, and we're gonna have this discussion, you and me, about honeybee viruses in general, how infection happens in honeybees, and then at the end, when we have the base covered, we can go and talk about honeybee queens specifically. So, do you mind to give us an overview about honeybee viruses?
Sure, why not? But before that, I would like just to thank you for inviting me, and also all those people who are watching us right now and later on, because I know beekeepers are busy keeping bees. It's the beginning of season here, at least in North Carolina that I am. And beekeepers are busy just installing packages and taking care of their colonies. So going with an overview, let me just share my screen with you, and then we will have a little bit of presentation, and then I can just explain it better. Can you see my screen?
Yes, I think that your screen is here. We can see it.
And what I can say about the viruses in honeybees, it's that they are a largest class of honeybee pathogens. So far, around over 30 different viruses have been found in honeybees. But they are classified in different groups. The big group that we can classify them are DNA viruses and RNA viruses. We have only one DNA virus in honeybees that is not dangerous for honeybees, but beside that, we have RNA viruses that they are the most problematic for honeybees. And among all those that are classified or grouped in different groups, we have three important viruses called the deformed-wing virus, sac-brood virus, and black-wing cell virus. So these three viruses have been found in most of the colonies, and they are specifically with the deformed-wing virus that we cannot talk about deformed-wing virus without Varroa mite. And this association between this virus and Varroa mite is #1 problem in honeybee industry worldwide, except Australia and few other islands. But beside those few islands, the deformed-wing virus and Varroa mites are the biggest problem in honeybee industry. Sackbrood and black queen cell virus, we can mostly see them problematic in brood and larvae. But then beside those, we have three most important acute bee paralysis virus, Kashmir bee virus, and Israeli acute paralysis virus. Kashmir bee,
Dr. Amiri, just 1 second.
yeah.
I think getting some. Uh, I don't think people are being able. You're probably going over your slides. I don't
Okay.
think the slides are going. Let's try again. Let me try to do from my end here.
Do you want me to stop sharing?
Because yeah, I think it's not working. Just let me just add this part here,
No, okay, that's fine.
and I think can work better. And you know to move forward or not with the presentation. So is this initial? Yes. Okay, let's start over. Sorry about that.
No, that's fine.
Okay. So this is the largest class of honeybee pathogens that you were talking about.
Yeah, if
DNA.
you click 2 more clicks, 1 more click.
Here you go.
I did talk about, okay. Then we have, did you hear what I talked about those 3 first viruses? The forming virus, sackbrood, and black queen cell virus. And then we have other acute bee paralysis virus group that are acute bee paralysis virus, Kashmir bee virus, and Israeli acute bee paralysis virus. So these viruses, we see them not in high level in the colonies, but still time by time we see them in the colonies when we screen the colonies for these viruses. But when they come and attack the colony dies very quickly. Then we have chronic bee paralysis virus. That is, I mean, beekeepers have seen it time by time, but not every year. It's just see it periodically. Some years they see it, some years they don't see it, and nobody knows how they turn from cover to over. That's one thing. And then I will talk a little bit more about it in a few seconds. And then we have a slow bee paralysis virus. So, and it's very rare. I mean, not many information is about a slow bee paralysis virus. But what brought me to be, I mean, what made it interest for me is that, as you see, we have, for example, we have a slow bee paralysis virus, and then we have chronic bee paralysis virus, and then we have acute bee paralysis virus. If you see these 3 viruses, you see they are paralysis viruses. But the thing is, for example, a slow bee paralysis virus, when it attacked the bee, it just from the infection until the symptom, it takes like 9 to 10 days that you see the symptom. When it comes to chronic bee paralysis virus, it's just 5 to 7 days from the time it infect the individual until you see the symptom. It takes like 5 to 7 days. When it comes to acute bee paralysis virus, from the infection until you see the symptom, it's very quick, 2 to 3 days, and then kill the individual, or like I will talk about it, how it kills the colony. So if we go to symptoms, beside those viruses that I talked, and they are found like long ago, since 2011, with new technology that we have right now, and next generation sequencing and bioinformatics tools, there have been other bunch of viruses that have been detected in honeybees worldwide, different countries. But the only thing that we know right now is a part of their genome. We don't have much information about the symptoms, about the virulence, and all about those things. We don't have, except Lexini viruses, those group of Lexini viruses. We don't have much information about the rest of the viruses that found through this, but mostly our information are a little bit more about the viruses that are listed here. So if you please click one more, you see here, to see the symptoms, because when I talk about the symptom, I see you in the left side, it's a chronic bee paralysis virus symptom in one individual bee. And with chronic bee paralysis virus, we have two types of symptoms. And one type that you see here is the bee gets shiny and the hair, and she loses the hair. And then still after a few days, she can fly, but she is nibbling by other workers in the colony, and then a few days later she dies. Or the other symptoms that we see with the chronic bee paralysis virus is the abdomen is bloating, and then it's just the bee is shaking abnormally, and you see the wings are like a car shape. So bees cannot fly, and they just move out of the colony, huddle in front of the colony, and then in the bottom of that individual bee, you see a bunch of dead bees, and in a few weeks, like in 2 or 3 weeks, the whole colony just collapse, go down, the population just collapse kind of, and then leave only a queen and a few worker bees behind, but the whole colony collapsed. So that is about chronic bee paralysis virus with the obvious symptoms. The other virus that everybody knows it's the form ring virus that you see in the right side, and mostly that is need to be noted that when I talk about chronic bee paralysis virus kill the colony or collapse the colony during the winter, when it's the mid of summer, when the population in the colony is the highest. But when it comes to the form ring virus normally kills the colony during the winter, and that's why we have winter mortality. So this is one abstract of viruses that I want to say, Humberto. Then it's interesting for us, if you want me to continue, is it's important for us to know how viruses transmit from individual to individual or from colony to colony. If you please click one more time, and you assume that we have 2 colonies, a beekeeper has 2 colonies, and then in the right time, May or a little bit later May, the colonies are swarming. So if the colony is infected by viruses and swarm to the next colony, so the next colony will be infected by viruses from the original colony. But at the same time, when you are doing beekeeping, and we know all that we are sharing all the equipment among the colonies that we have in the apiary. So a beekeeper, by sharing the combs from one colony to another colony, if the colony is infected, you can just easily transfer the viruses or transmit the viruses from one colony to another colony. And specifically, early in the summer or early in the spring or later in the season, when we have some few colonies that they are not as strong and we would like just to get rid of those weak colonies or queenless colonies, we just combine them together. And specifically, I'm not a fan of combining colonies later in the season, because if the colony is assumed the colony was queenless for a while, and then all of the bees just emerge and they become older bees, and so there is no any brood in the combs, and all the Varroa might just come to the older bees, and then easily you add this colony, a few frames of older bees in top of another very strong colony that you have. It means that not only you are transferring some bees to the new colony, but you are transferring diseases and all those the Varroa mite and all those pathogens to the new colony. So I'm not a fan of combining small colonies later in the season with the big colonies, because not only you are combining the bees, but you are combining the virus, and you add so much pathogen to the strong colony. So that is the case. So these are the way that virus can transmit. Even virus can transmit in the habitat when the bees from, if you click one more time, please. if bees from your neighboring come to the habitat around your colonies and they are deposit the viruses, there are a bunch of studies that shows that viruses can be deposited in the flower, and then when the next bee is coming to collect nectar or pollen, they can just harvest viruses as well. But assume, as I did talk about this later season, we always, and we, as a beekeeper, we have seen robbing. If you click one more time, and when normally the weakest colony is robbed by other bees. So it means that weaker colonies have higher level of pathogens and viruses as well. So when bees are robbing one weak colony, and they are taking every honey from these colonies. So not only taking honey, but also they are taking viruses to their own colonies, to their own colonies as well. So this is the ecology of the viruses outside of the colony. When you go inside the colony, viruses are transmitting in different ways. For example, older bees, when they are feeding the larvae, click please, they are transferring viruses from their hypopharyngeal gland. That viruses are going, and then they add them to the larva food, and they make the larvae infected. When they are sharing the food among themselves, or they are feeding the queen, they are transferring virus from every individual to another one, or from worker bees to queens. But just imagine that this has been the story from very beginning, that the viruses have been around without any problem. Even though there have been there are some studies showing before Varroa mite we have had a colony death and some of the problem, specifically with chronic bee paralysis virus or acute bee paralysis virus. Mostly those studies reported from England when the older generation were there, and the first, I think the first virus lab was established over there. But then after Varroa mite came, things has changed. One click, please. And the Varroa mite is just a game changer, Humberto. Yeah, the Varroa mite is just a game changer, and not only increase the level of viruses in the apiaries and in the apicultural apicultural industry, but also directed some of the viruses toward more being virulence, like the forming virus that we have good amount of evidence showing that because Varroa mite is not only the mechanical transmitter, but also biological transmitter, meaning that when she gets the viruses, specifically the forming virus, replicate in her body, and then she just transferred that virus to the next bees that she is infesting. So she become kind of problem in the honeybee industry. So this is so far that we have. So Humberto, if you want, I can just talk about a little bit of, so this is a basic we made. Before going to the next slide, if you want to say, okay, people will ask, so far so good, but what is the connection between all these viruses that you talk with the honeybee queen? Then I would like to talk a little bit about, if you have.
Let's try to. There's some echo going on. Wait a second. I think I need to remove this.
Yeah.
Hello?
Yeah. Can you hear me now?
I can hear myself for some reason. Okay, I think we're back to normal. So I think I want to recap and just to a little overview. We have two separate stories here. One before Varroa mite, and one that we're gonna talk more now, which is after Varroa mite, and we're gonna include honeybee queens in this story because as you guys are gonna see at home, there is a big change in how viruses are transmitted and how the whole cycle changed from before Varroa mite to after Varroa mites. Until now, we cover what happened inside the hive, what happened in the environment, how the virus is spread without Varroa, implications of Varroa, without Varroa injecting the virus directly in the hemolymph or in the body of a honeybee. And that could be a worker, larvae and pupa, and could be also a drone.
Yeah.
Which, by the way, which is where Varroa prefers. So, and that's the story start to become much more interesting, because when things start to reach the drones, the reproductive system of a honeybee hive, things start to become more dangerous. And I think now we're going to enter it more in everything that Dr. Amiri is studying in the last 5-7 years, if I'm following you correctly. Several publications on that. And if you can give us a little overview about the last 7 years in this research field, we will really appreciate, because the story, the problem just started when the Varroa came out and started to infect the reproductive system of honeybees. And that's where the danger started, right?
Yeah. What I can talk about a little bit of about Varroa mite and how Varroa mite changed the things. As I said, there are different publications, very strong publication. There's the story about the Hawaiian island that you know about the Varroa mite arrived. There's story about New Zealand that Varroa mite just came, and then it started in the north and then moved toward the south of New Zealand, and how things changed quickly and dramatically. And with the matter of how the virus level just increased a lot, and we have pretty strong evidence that Varroa might change the story from, let's say, people thought that viruses are just blind, they are not problematic, they are okay. Bees can handle them, and for a long time, bees develop antiviral defense mechanism. And then when the Varroa mite arrived, things changed completely. So then the viruses become problematic, and they become a risk factor for honeybees. So that is the thing. And then when it comes to honeybee queen, people say, well, Varroa mite is not infesting queen, and that is not the problem for queen. But that is not the story. That is not true, because Varroa mite, first of all, is interested to infest drones. That is what we know. That is the fact. So when Varroa mite is infesting drones, she is transmitting so many viruses to drones and make the drones infected. So and these drones, most of these drones, some of the drones become deformed, and they cannot fly. So when they cannot fly, it means they cannot mate any queen. But many of them still can fly, and they can compete with the queen. I will just go to detail with that, because we did a study on that. I will just go toward that. And then they can mate with the queen, and they can compete with the queen, and they can transfer the virus from drone to queen, so, and make the queens infected.
Do you want me to stream again the slides? You let me know.
No, that's fine. I will talk about it later. But just one overview. The other story about Varroa mite, because Varroa mite is infesting all the worker bees. When she is infesting the worker bees and transmitting the virus to worker bees are they are covert symptoms and overt symptoms. When we talk about the overt symptoms, we see deformed wings, deformed wings bees. But covert bees, and we know most of the viruses are coming to the hypopharyngeal gland, and bees are not able to produce enough royal jelly when they are feeding the queen larvae, or if they are feeding, the virus is transmitting to the royal jelly. So first of all, if you haven't done, I did it. I grafted queen larvae to produce queens in healthy colonies and highly infested colonies. And I have the project is running right now. The result will be ready in short, like in a few months. And interestingly, when you have the grafted queens in the infested colony, high level of Varroa mite infested colonies, you see that there in each cell there is not enough royal jelly. And then when comparing these groups with the group of healthy inside the healthy colony, you see because they don't get enough food, they develop quickly because they don't have food. They don't stay to finish the food or royal jelly and then go to capping. They go to capping quickly. So it means that they are develop very quickly and they cannot complete those systems that they have, developmental process that they have. So these are the things. And then out of those colonies, I mean, I got queens out of those colonies with highly infested. I terminated the experiment before capping or right after capping. But I'm sure that these queens are not going to be high qualified queens. High qualified queens. So that is in the colony level when you see it. So, but if you want me to explain a little bit more of details, please just share the screen, and then I will talk specifically when the queen become infected. And most of the time we see the first one is fine, but that's okay. The first one, if you have a queen, infected queen, she is producing infected eggs, and we have pretty good amount of knowledge around it that viruses, specifically the forming virus, sackroot, and black queen cell virus, they are transmitting vertically. Vertically means from queen to the next generation, to the eggs. So, and we have found viruses, these three viruses, and other people have also found viruses in the egg. So now I'm not talking about the pathogenicity of the viruses afterward, but I'm just talking how the viruses can transmit, and then possibly if these eggs become queen, they become infected from early in life. But then we have, when they turn to be larvae and they are fed by worker bees as a royal jelly, and you see they are eating a lot royal jelly. So if the worker bees are infected, assume they are infested by Varroa mite, they are infected by the forming virus in high level, and they are producing royal jelly. And this royal jelly is full of Varroa mite and other viruses. And it's just the larvae is eating all this royal jelly and ingesting so many particles of the viruses. And think about sackbrood and black queen cell virus here, specifically during the larvae and when the larvae is eating viruses along the royal jelly. And if the amount of virus particles increase, I'm sure as a beekeeper you have seen it, that they die, and later on when you look at the cells, you see bees start cannibalizing those larvae. I'm sure that you have seen it. So right after emergence, we have two steps here. We have larvae, and then right after emergence, the queen will stay in the colony for a few days, for at least 4 or 5 days, 5 days before flying for emergence. And this is the time that the immune system of the queen is completing. It's just developing. And specifically people saying that specifically 2 days after emergence, the bees and the queens are the weakest in the immune system. And assume, that is one of the reasons that queens stay in the colony for 4 or 5 days before flying for mating, to have the more immune the immune system become more mature. And then because when she is flying, she flies for mating, she is by herself. No any other bees can take care of her. And that's why she needs to be strong in immunity. She should be able to fly. She should be able to take care of her during this 10 to 15 minutes of fly collecting a sperm. And assume that those drones that I talk about, and they are infected by viruses, and they are just coming to the drone congregation area and mate with her. And not only they are passing their sperms to the body of the queen, but also they are passing the viruses to the body of the queen. So then she comes back. She comes back to the colony and stay in her colony for a long time. Sometimes 1 year, sometimes 2 years. I mean, very rarely nowadays with 3 years, but 2 years, that's fine. And she is in contact with all the worker bees that are either infested by Varroa mite, either infected by viruses, or those worker bees that are somehow infected by viruses, and they are feeding these queens, and they are touching these queens, they are antenning these queens, and they are feeding these queens. So they are just passing some of the viruses to her body, and that is why you see when the early queens, if you take care of everything from very beginning, later when we survey the queens, we see more virus and higher level of virus from the inside the body of the queen. And that is important. That can be either she is receiving more virus, or because of age, she doesn't have that strong antiviral immune system. So the age can cause a little bit of the immunity weaker in her body. So that is the open question that we need to study and understand it completely. Do you want just have question, Humberto?
Yes. Wait a second. Let's get some questions from the people at home. So if you guys have questions, please let us know so we can start. Don't feel shy. That's the time to ask questions. I think that helps myself and Dr. Amiri to situate ourselves, because it's too much information that we're giving here, and sometimes it's better if we have some interruption and see how the people are assimilating this new knowledge. So let me know if you have questions. If you don't have questions, we can continue the talk here. So I don't see any questions right now.
What I can talk about, Humberto, while people are just writing in a chat or so, it's the pathway of viruses. We have a pathway of horizontal transmission that we talk about. Different pathways are they coming, and this is the way that virus transmit from the same age bees or from the bees to queen, either feeding, either like Varroa mite, or just passing vertically from one generation to another generation. And what the evolutionary studies are showing us, that when the virus is transmitting vertically from mother to next generation, it becomes low virulence. And it's because the virus by itself, I mean, there are different mechanisms, but the virus by itself, or either queen just choose to have the low virulence viruses, or virus just try to be more benign, because virus wants to spread in the population. So the queen is infected by virus, and the virus want to, if the virus kills the queen, then the life of the virus is finished by that. But if the virus stay as benign and going with low virulence, the virus can pass through and go to the whole colony and spread to the whole colony. When it comes to horizontal transmission from bee to queen, from bee to bee, from bee to larvae, or through Varroa mite, it becomes a little bit more acute side, let's say, and more virulence that kills the individual in the colony. So that is the case. Even venereal transmission. When we talk about venereal, it means through the mating or sexual transmission. Still, even though it is venereal, they call it venereal, but still it's a horizontal transmission, because queen and drones are from the same generation. So, and it's like when you talk about, I remember in one of your clips or video clips, you talk about Varroa mite and how viruses become more virulent when the Varroa mite is transferring, and because it's injecting so many virus particles to the body of the worker bees. And it's almost the same story with drones. When they are injecting, they are mating, they are injecting viruses to the body of the queen, and these viruses become kind of more virulent than that. And that is another things that we need to take care of, that before Varroa mite, maybe that this mating or transmitting virus from one generation to another generation, the mating was most important route or pathway for the virus to transmit from drones to queen and then to the next generation. So that can be explained in that way as well.
The mating flight was always something that got my curiosity about the honeybee biology itself as the social organism, because for a long time, it was hard for me to understand how evolution would select such path that creates so much danger for one single individual that can, if the mate doesn't happen, the whole colony die. And she go after, expose herself to nature and all kind of conditions. And in the middle of that, she need to mate with 30, 40 very healthy drones. And so there is so much that need to happen at that time. And she come back. So, for example, here in Florida, we have a problem with dragonflies. Dragonflies love to wait for these congregational areas and just not only eat the drones, but also eat the queens themselves. They really love them. So there is so much exposure and dangers. And now, on top of that, we have a whole virus system that apparently evolved to take advantage of that reproduction system to not only infect a couple of bees and kill a couple of bees in a hive, but infect the reproduction system of a honeybee, of the colony. And in one shot, he spread to everybody in one single shot. It's very amazing to me to see how nature works sometimes in this situation. So I'm going to start to stream again your slides so we can move on with this conversation.
So if you move to the next slide, please. So as I said, these venereal transmission, and it's because let me continue a little bit of your discussion, Humberto, because for a long time, beekeepers thought that, well, drones are not important because always you see them in the colony. They are not, they are there, when they are there, so they are cheap, but that is important to know. And now we understand the important part of the important role of drones, because the whole life of queen is depend on drones.
Uh-oh. Something's wrong. Please let me know in the chat if you guys are still here. I'm not sure what's going on. It's maybe something with the connection of doctors.
I don't know what happened.
Okay, you're back. Great.
I am? Okay. So this is the technology part of it. It's not.
Well, that's just another proof that we are live.
Yes. No, the thing is, the whole life of queen is depend on drones. So it means that beekeepers, specifically those that are, or queen producer, let's say those that are producing mass number of queens and they are spreading to the rest of the country, they need to take care of drones, and I'm sure they do. And they need to have healthy, strong drones that mate with the queens and have higher quality of queens. But here, what I want to talk about is one of the studies that I did during my PhD when I was in Denmark. And the good thing about this study is using the system or queen production system or queen mating system that exists in Denmark. And because there are a bunch of, let me just talk a little bit different from the other angle of it, more broadly about breeding, not only in honeybees, but in the animal as well. When we talk about the breeding, it means that we need to select the right parents. After we selected the right parents, we need to have the method for mating. Is this selection and mating needs to work together to have a high quality of progeny for next generation. It's not only about the honeybees, it's all about the other animals that we do. But with honeybees, it's a little bit complicated because of the nature of honeybee queen mating behavior. And because when I talk about Denmark, and I'm sure many other countries have such that facilities as well, there are a bunch of small islands around the country that beekeepers utilize those little islands for mating, as a mating station. It means that they can select for the mother line and have the virgin queens to those little islands, but they can select for the father pattern line as well, the father side, and move those colonies for producing drones to those islands as well. So it means they can select for father and mother as well and move them to that island. So in that island, because they are a little bit far from the other islands and the main land of Denmark, so drones cannot cross the islands. It's just the drones that are available in island can mate with the queens. And this study was done in Denmark. That's why I want to explain it a little bit and just let you know that we utilize that system. So, and then if you move to the next slide, please.
I got just 1 second here. I just want to make sure of, I need to remove this for a little while, just so I can, can you hear me?
Yeah.
Can you hear me well? Okay.
Yeah, I can.
The thing, if I understand correctly, so the island that you work on, just to clarify for myself and also perhaps to some people at home, is the only way that we have to control the petri line of genetic selection of honeybees, correct? Because one of the ways.
One of the ways, let's say, because right now we have insemination.
Okay. Without taking insemination, but in natural conditions, we need a separate place that we know for sure that we can control the drones, correct?
Exactly.
Because if you cannot control the drones, which happens in a lot of queen operations that I know here, that people claim they have genetic stocks and they can keep in certain conditions, and I'm always trying to argue that you cannot control the drones, but so there is a dilution of whatever you're trying to select. There is always a dilution of that specific characteristic that you wanted to select. But in your case, you were working in a very restricted place, an island where you have full control of all kinds of hives that you have there, including the drones. Am I correct?
Yeah, you are completely correct.
So that means that the study was done in a correct environment. That's what I want to be very clear to the people at home, because there are many studies out there trying to do similar things, but because they cannot control the drones, I always arguing with those researchers and beekeepers about that.
Yeah.
Let me continue here. All right.
Yeah. So then what we did, we were not the first people studying this transmission of viruses from drone to queens, because before us, there were a bunch of other studies. Actually, this idea of different pathways that queen can be infected by viruses, I think it started by Judy Chen and the group by 2005 or 6. They collected drones, and they have such that nice graph still available. And then later on, V. Miranda and Ingemar Fresh continued with that. You and Gresh continued with that. And then people tried different ways, specifically with the artificial insemination, to understand the virus is transmitting. And like, I think 2 years before us, it was Orlando Llanes that did, and they just collected a bunch of drone in the drone congregation area, and they collected queens after after queens started laying eggs. And I want to just to give them credit. But the only part that was not answered, because this mating behavior was kind of mysterious for everybody. And that's why we wanted, and because that was not clear, okay, if all this happened, how does it happen in the air? And that part, that little part was not answered. And that's why we wanted to answer
Yeah,
that.
there was a lot of assumptions on those previous studies, but nobody was able to demonstrate.
Yeah, exactly. So if you
And,
go please to the next slide,
and here you go.
and the next click, please. So then we had three questions. The first question is, first of all, because previous studies showed that the queen is flying to drone congregation area, and those infected bees infected by the forming virus are in the drone congregation area. Are they able to compete? These drones are able to compete with the healthy drones, because that's this what we understand. We are not completely sure, but what our understanding is that because these drones are willing to pass the genes to the next generation or to the queen, they are competing to do that. They are not just friendly mating. They are competing by themselves. It's thousands of drones that competing to mate with the queen. So do these drones have enough energy to compete and fight with other drones to be able to mate with the queen? That was the first question that we had. And then the second one, if you click, please, we had how risky that mating, if she mate with that wrong individual or wrong drone, how risky it would be for the queen to be infected. Or if these two are happening, third question. If these two are happening, like infected drones are able to fight and mate with the queen, and even they are able to pass the virus to the body of the queen, and we know that all these sperms are collecting to the spermatheca. They are safe in the spermatheca. And is there any mechanism in the spermatheca that not only keeping the sperms, but also keeping the viruses inside the spermatheca, that doesn't allow the viruses to pass to the next part of the bodies? These three questions, we had them in mind, and we wanted to answer these three questions. And that is why we had one single mother queen. If you go to the next slide, please. We had one mother single queen that we tested for all the viruses at that time. And the beekeeper, we worked with the beekeeper, very careful beekeeper. And we had sampled worker bees and larvae from these queen. And even we had the nurse colony that later on the beekeeper grafted and kept the grafting larvae in that colony. We tested them and we found that these two colonies are healthy. And that's why he produced bunch of sister queens. And after grafting, few days later, because as a scientist, we are checking every step and understanding when things are happening during this process. When the larvae started to grow, later on we had 15 of these cups with the royal jelly and larvae in them as a sample to be checked that, okay, we already checked the nurse colony. We wanted to be sure that the nurse colony is healthy. And then after the cells were capped, mostly queen breeders just move these capped cells to the incubator, and they don't, after emergence, these queens don't touch any bees until you introduce them to the colony or mating nukes. So we sampled 10 more queens from there, but the rest of the queens, we introduced them to the mating nukes and had them in the island that we provided infected or not, let's say infested colonies by Varroa mite, that for the last 3 years they were not, the colonies were not treated by Varroa mite, for Varroa mite, and they had high level of Varroa mite, and we were sure that these drones from these colonies are highly infected by the deformed wing virus. But what we did, because still we wanted to control the environment, that these queens are not being infected by bees that are bringing infected pollen to the colony, or in another way. In eight of these queens, we introduced them to those mating nukes and had the queen excluder in front of the nukes. So and then later on, we treated them 2 times with the CO2 to stimulate laying eggs. So they did not mate, but they started laying eggs. So that's what we can. So if you go to the next slide, please. With those queens that we introduced them and we wanted to be mated with the drones, we had this queen excluder in front of the nukes, and every afternoon we were going and checking when the queen is coming behind the queen excluder. As soon as the queen was behind the queen excluder, we removed the queen excluder, did let the queen to fly, and waited until the queen is back from mating. I see it's not, okay, now it's, and as you see here, with the queen excluder, bees can pass through back and forth and can bring pollen and nectar and so on and so on, but queen cannot. And then after she is back from mating, normally she is back with the mating sign or the endophallus from the last drone that she was mated with. So she brought that sample to us. And then normally, this is the job of worker bees to remove that mating sign or the last endophallus or the endophallus from the last drone that she was mated with. But here, because we wanted to check those drones that she did mate with, and that's why we collected these mating sign or the endophaly from these queens as a sample, as you see here. And we collected that. So these queens sometimes did mate once and they flew only one time, but sometimes they flew 2 or 3 times. And every time they came back, they brought one sample for us. And then later on,
Dr. Amiri, let me just show this one more time because I think this is beautiful. So very quite interesting. So you block it. So your sample was exactly what happened on a mate flight. Was not assumption. Was not collecting random drones here and there. You collect whatever happened in real life there, which is a very ingenious way to do it. Yeah, very, very nice.
Yeah, I should say thank you to my supervisor, Pierre Keroge, that lately I will thank him as well, that he had that idea. And because I was a beekeeper for some times, I was not afraid of bees, and I was just working easily in the field, I could just run the experiment in this way.
Yeah, this is beautiful because there is no question about it. You collect the perfect sample, I would say. It's the perfect sample. Whatever happened in the air, naturally, you were able to capture that back.
Exactly.
Very nice, very nice. Good work.
And then we did let the queens to go inside. And because, if you just right away, just dissect the queen, just sacrifice the queen and dissect, you are not sure that the virus will be localized and start replicating in the body. We did let the queens to go inside and started laying eggs for 3 weeks. And after 3 weeks, we collected the queens and dissected them.
So I have a question for you. It's a research question, actually. So in this case, you get the sample from the last drone, right?
Exactly.
So do you ever thought about the other 20 drones that was there? What kind of random sampling that you might have getting with the system? I'm just curious if there is any way to.
I mean, we did not collect any. Of course, we did collect drones that coming back to the colonies, drones that are coming back from after later in the evening when they are coming back from drone congregation area to their own colonies, and we sampled and we did check them for the viruses, and that was like the part. But we don't know about the other 20 or whatever number of drones that did mate with this specific queens or with every other queens. You know.
But but it's quite interesting. But
Yeah.
once you find something, you know it's the real deal.
Yeah.
Cool.
Exactly. So if you go forward, please. And that is very beautiful. Even though many beekeepers, I mean, I was a beekeeper for like some years, like 6 years. Never I thought that I can't see the queen comes with the mating sign, and I said no. I mean, first my supervisor was offered that kind of a study to me. Said, no, that's not possible to see these queens, you know. But then he showed me how to do it
Yeah,
and how
you
to get this.
crazy researchers.
I know. Yeah, I need to thank him. So you see, and then after three weeks, we collected the queens and dissected them, and we wanted to see. So if the queens receive the sperm and the virus, is the virus is spreading inside the body of the queen, or it's just inside the sperm and spermatica? And that's why we did dissect the queens. We collected as a head, thorax, and then abdomen. We opened up abdomens. We got the ovaries, as you see in the picture. And then even we got the sperms out of a spermatica. We wanted to check those, and the spermatica, what remains back. And then
The sperm and the spermatica.
and the spermatica, yeah,
Gotcha.
separately. And then we did test them in the lab for deforming virus. So if you go with the next one, please. Let's just go back and think about the whole project. So
Oh,
back.
you want to go back?
Yeah.
Okay.
Yeah. As you see here, let me talk about different colors that you see. Let's just review what we did talk. We talk about that mother colony, and we talk about the grafting and sampling throughout the developmental stages. We collected larvae and royal jelly. We collected newly emerged queens, and those queens that were introduced to the mating nukes. Some of them did not get a chance to mate, and some of them did. And then you see different colors here. When you see blue, for simplicity, I can say, when you see blue, or at least in my screen it's blue, it means that we were not able to detect the virus. Okay. When you see light green, it means the level of virus was very low, less than 1,000, let's say, particle of virus per sample. When you see orange, it means above 1,000 particle, but below 10 million virus particles. But when you see red, it is above 10 million or above 10 million virus particles. And here, as you see from larvae and royal jelly, most of the samples, except for that, very low, most of the samples are completely clean. No virus was detected. So it means that the nurse colony was healthy, as we saw, or at least we were not able to detect any virus. When it comes to newly emerged queens, we dissected them to head, thorax, and abdomen. Still, the same story is going on. Not much virus, except one of the samples with the thorax and abdomen, and one another sample with only thorax. But the rest, either we didn't detect any virus or very low level of virus. So, but when it comes to the queens that we introduced them to the mating nukes, if you go to the next slide, please. So first of all, let's talk about those 8 queens that we did not let them to mate. We had them, we exposed them 2 times to CO2, and they produced only eggs without any sperm in the body. And we had them head, thorax, abdomen. We had the ovaries out. We had spermatica out, but because they did not mate, they didn't have any sperm. So as you see, most of the or majority of the samples are still clean or in low level of virus. Okay, so the environment and all the process that we checked about the process time, nurse colony, and the environment that these queens were in. And then if you go next, let's talk about those samples that we collected, okay, those spermatica. Some of the queens we did not get any endophallus from, or as you see, they are white or NE, no endophallus. And some of them with the green that you see, low level of virus. Majority of the samples, those endophyli that we did collect, were orange. It means that the drones were infected, and 3 of the viruses of the samples were highly infected with, and you see it's red. So they were highly infected. So, and assume this is only 1 or 2 drones of those drones, 20 or whatever number of drones that mated with the queens, and the rest we don't know about it. So if you click one more time, please, and this is the number of queens. We had 30 queens were infected in this situation. And after 3 weeks, we had 3 groups of queens. One group in the top that you see, 7 or 8 queens, they did not develop any virus. So that is what's kind of mystery still. Either they did not mate with the infected drones, which is not true, which even one queen got mated with highly infected drone, and you see it's red, but we could not find any virus in the rest of the body or very low level of virus. Majority of the queens developed virus in medium level, let's say orange we see. And the rest of the queens, you see them as red. The level of virus increased a lot, even in a high level of virus. Even one queen was dead, okay? So after mating, she was dead. I don't know exactly if she died because of the virus or she died because of something different. But when we did test her, she was infected with virus. So this is how it shows that the virus from mating can transmit from drones to queens and make the queens infected by the forming virus. And why we did test the forming virus, let's talk a little bit about that, because the forming virus is a good model study that you can find it in all the castes of honeybees, and you can in drones, workers, and in queens, and also you can find this deformed wing virus in all the developmental stages. So because this is a good model, we wanted to show that the viruses can transmit from drones to queens. It means that the queen producer or beekeepers need to take care of drones, need to have healthy drones to have healthy queens. If you don't care about drones, then you will not have a good quality queens. That's the main message here for the beekeepers.
Yep, that's a very simple. You demonstrate that's very easy to see, especially after all these footages and results. If you don't take care of the drones, you're gonna end up with bad queens, no doubt about it.
Of course. So this is the main things, that main route that the queen can be infected by viruses, and we demonstrated in a very careful study showing that. So this is how virus transmit, and most of the studies that have been focused on honeybees are mostly about how virus transmit from drone or workers to queens. And that is important because of the way of transmission and virulence that I talk. But believe me, it's very difficult when it comes to the queen to study the pathogeneticity or pathology of the viruses. Because the queen, even though she is one single important individual in the whole colony, that the health and vigor of the colony is depend on her, but she's only one bee inside thousands of bees. And it's difficult. And most of the cases, we see dead queen outside of the colony. Okay? Or, as you see in the picture, we see it while developing. If they are infected, in this case they were infected, bees are removing them before they emerge.
Yes.
Or they are dead, okay? Or you can see the dead bees. I mean, the other two picture, I purposely infested them by Varroa mite. And you see one, the middle one, and the Varroa mite is still there, okay? She emerged, but she was dead. The other one, she emerged, but she did not have wing. So when she doesn't have wing, she cannot fly, and she died few hours later, like a bees, like a worker bees. So this is very important. And that is why most of the studies on honeybee queens have been focused on the transmission routes. And less and less we have on the pathogens and pathology of the viruses in honeybee queens. But we have some studies on the pathology of the queens. We know that, for example, in nature, black queen cell virus causing pathogenicity to the queen larvae pupae.
In the larvae pupa stage. Yeah.
Yes. Okay. We know about, I myself studies chronic bee paralysis virus. Most of the studies that we have done is in the lab or with the old queens. I will talk about the old queens now, and let's say failed queens or healthy queens. I will talk about it. But I did talk about the chronic bee paralysis virus. I did inject them or applied virus in top. And then few days later, you see the abdomen is started to be bigger and bigger. And then after you open the abdomen, and then queen is shaking, she cannot fly. Even I did test the same story with the Israeli acute bee paralysis virus, the same thing. And after you open up the abdomen, you see full of hemolymph and all those pathology effect of the virus. But when it comes, because most of the viruses are at the covert stage in the queen, so we don't see the right symptoms. When we bring the queens in the lab, we do test them with different methods, and we say, okay, this level of virus, that level of virus, this virus, that virus, that virus. And recently, even I am part of that study, that this study showed that this covert infection can cause the lower ovary mass of the queens. And that's why some part of this queen problem is because of the viruses. And
so meaning when the viruses are infecting the ovaries, they produce less eggs or defected eggs or something like that?
We don't know that, but right now we know that the ovary mass is lower in the failed queens that had higher level of virus than the healthy queens that had lower level of virus. This is what we know. We don't know the production level. We don't know, because even we did a study here in Greensboro, and we did infected the queens with the Israeli acute bee paralysis virus, and we wanted to test how worker bees are accepting these queens and how they come to attend the queens. The study failed somehow. Okay, and it's because it's difficult to work with queens and workers in the same time out of that complex environment in the colony. But what I see right now is moving. The more and more interest is coming to that, to understand the effect of viruses, not only the transmission route, but the pathology of the viruses on the queen and queen production, specifically with the ovaries, the effect of viruses on ovary, and the effect of viruses on spermatica, because when the queen become infected with viruses, she is in challenge with virus, and it takes part of the energy from the queen to keep them down. So she needs to remove some energy from the other parts and bring them to challenge with the virus or fight with the virus. So
That,
this is
not only that, fighting the virus take energy from her, and the virus replication itself consume resources that were supposed to be doing other things inside the honeybee queen that now is going to be producing viruses, and that also put the queen, all the resources inside the queen going down. Yeah, viruses are tough. Viruses are tough.
So by now, before just you move to the other level, would you please just to go to the next level, to the next slide? And I would like to thank my supervisors and advisor during my PhD, Pierre Keroge and Marina Meixner. And also, I would like to thank Dr. David Tarpey and Dr. Olaf Ruppel and the groups that I am belong to, because I'm working with both of these great scientists and the group that supported me during all these years. And plus, my funding agencies from different resources that supported my research. So I'm happy to answer any question that you have, Humberto, and all the people who are watching.
Yeah. Nice, very nice. Where is Oliver Ruppel is here?
Yeah,
where is David? Oh, here he goes.
David is down there.
And, and this is your.
This is Pierre Keroge.
Yeah, I don't know him in person. I read a lot about him, but never met him in person. And all the agencies, the funding agencies here. Yeah, very nice. Amir, thank you. I think it's the time, if you guys at home have questions, I think that's a good time to start to write those questions in the chat room if you have them. But I have questions for you, and basically it's some curiosity that I have when I'm in the field with commercial beekeepers, and especially queen breeders and queen producers. I see a lot of royal jelly coming from everywhere in the world, where they use the royal jelly to prime before they start to do the drafting. The grafting. So they prime those queen cells with royal jelly from many different places in the world. And I was worried about, if the royal jelly from these different places could be bringing new pathogens, and your thoughts on that.
Humberto, this trading is causing a problem. Not only royal jelly is coming, but also think about wax. Think about pollen. Pollen is just very easily trade here and there. And one way people thought about X-ray them and this just kill the pathogens in them. I don't know about the royal jelly, if that can be X-rayed or not, to kill the pathogens before using them, or how the cost of that process. But interestingly, I think there is another study that shows even inside the U.S., if you have bees, because bees and viruses are co-evolved together, and after some times they become like they can tolerate each other a little bit. But then you see, we move the bees from one side to another side, even though in the other side the virus has the same name. Let's say the foreign virus or Escherichia acutely paralysis virus or whatever, but it has the genetic side is a little bit different. Think about this COVID-19 that we are dealing right now with. It's a COVID-19, but now we are having different varieties from different countries. It's just, the name is same.
It's the same, yeah. I was about to do the link. I was preparing myself to do the link too, and you did it too. Yeah, it's exactly what happened with us right now. It's happening with the bees for the last 30 years now. We are observing in real time a coevolution of virus infection that came through Varroa mite, and it started a new cycle of selection. And we're not in equilibrium yet. We're still seeing those viruses popping up everywhere in different strains, because things didn't come up in equilibrium now. We're still living that phase where everything is happening and all these new strains, and then the bees need to come up with a new defense, and then the virus come up with a new approach and go on. So yeah, that's exactly what's happening with the bees, and we have the perfect example with COVID-19 right now.
Yeah. We have a new technology as well. Like I think last person that you had a live stream, Sean, they are talking about the new technology, but that is good. That is possible to do it. But the only thing is that these viruses are in the evolution. They evolve time by time. They change. They change. And that's why new technology, we need either to be updated very quickly to combat them, or just have the bees that are able to control them. We do, I mean, part of the story, it's part of the things that with us as a beekeeper or as a scientist to understand and take care of all these steps that we talk about. But part of it with honeybees. And honeybees, we need to select them to be able to cope with the viruses and diseases. And that's why you see with the story with Varroa mite and all this hygienic behavior and all the things that are going on, it's just we are trying to just going shortcut to reach that point that bees can control the diseases by themselves. So that is the story of with Varroa mite. So we need to have it with the honeybees as well, with the viruses as well. So that is all that we need to do, and I'm sure that we can do it.
Yeah, we need to help the bees the way we can. Selection is one way to help the bees to speed up the process so we can get an equilibrium faster. But it is really hard, especially for beekeepers that need to put food on their table and that depend on the bees. So sometimes to reach the point that we need to reach, lots of sacrifices need to happen, and it's not everybody that is willing to do the sacrifices, and that's where you're always stuck, I would say, in our own evolution to help the bees to reach that point. Yeah, it is a fascinating subject, and we can be here talking for hours. I have the stamina to do, but I don't know about you. So you let me know when you're good.
So, one more thing that I can make here. I'm sure that those queen producers, specifically those that are professional queen producers, they are taking care of many of these details that we talk about. But many other queen breeders or queen producers that we see, maybe they are a little bit careless, or if they are careful, but they are having their apiary or operation in the place that there are neighbors having colonies. That sometimes those are not careful, because when it comes to mating behavior and all this system that we talk, or free mating behavior system, then you are careful, but your neighbor is not careful. So it is like a collaboration that you make by yourself, and then you make the collaboration with your neighbor beekeepers, and then have them all in the same table, that if we want to have a healthy queens, we all need to take care of these details. Or just make an agreement with everybody in your neighborhood.
Very interesting subject. I'm not sure I'm getting the questions, or if people are shy, or I'm not receiving the question in the software that I'm running. Let me run to the YouTube itself. Just give me one. I want to make sure I'm not getting the questions. Okay, I think I can see people from Facebook, but I'm not sure I can reach the people on YouTube for whatever reason. Oh, on YouTube for just whatever reason. Second, and now I need to mute. Okay, are you guys still there? Sorry about. Duncan Mason is saying Jay Evans. Oh, he's just answering them all. Okay, Jay, thank you to be with us. But I think, wow, there is a lot of stuff going on YouTube, and I didn't, for some reason, I'm not getting this in the software. Okay.
Let me add one more thing here. You talk about Jay. May I?
Yes, go ahead.
Thank you, Jay, if you are with us. But I was thinking about mentioning your new publication and your new method.
Just
And
to
I think,
clarify to everybody, Jay Evans is the research former research leader at the Beltsville lab, the USDA Beltsville lab in Maryland. So just for people that don't know Jay.
yeah. The thing is, we did talk about the selection and mating, and one way for selection is to know that bees or the mother queen is resistant or tolerant to the virus, and they develop a system that you can inject, and they had like a green fluorescence with the virus. They integrated to the virus, and then when you inject the virus to the body of the larvae or pupae, the virus is not propagating. There is an antiviral system in the body that doesn't, and it's just give the opportunity to select the best and antiviral those that are tolerant or resistant to the virus, those bees. So I think they smartly developed that one, and I don't know how easy it can be used by beekeepers, but this is one method that you can select of those resistant bees to the virus.
Interesting. Okay, Dr. Amiri, I have a couple questions here from the YouTube people that I didn't receive. I don't know why. WR Farms is asking you and myself, how effective would be OAV, I think it's oxalic acid treatment, but during the nectar flow, be using a 12 treatment regimen. I'm not sure I'm understanding the question here. Every other day for 12. I think he's asking how effective would be a oxalic acid treatment for Varroa again if you use it every day. Wow, that's a lot of labor there, sir. But let me know if I don't. You can ask again here in differently in the chat, but I'm not sure I understood the question. 18 Bees is asking us, Dr. Amiri, when colonies have deformed wing virus during winter, do the bees leave hive to die or drop dead inside the hive?
Let me just clear this one beforehand. And there is a study that came out from Sweden, and they treated the colonies, and they wanted to see after they treat the colony for Varroa, how long after they can find viruses or the viruses are high. And they found out that the generation of bees need to change. So at least when you treat for the Varroa, you give it 2 months to have the healthier bees for specifically before winterizing the colony. So when you have the unhealthy bees during the winter, of course you will have the colony death or mortality, high colony mortality. So, but if you take care of Varroa, and Varroa will drop down. If you drop down the Varroa, then the viruses will drop down a little bit later when the generation of bees change, and then you will have a successful wintering, or you have a low winter mortality. But when bees are, if I understand the question right, when bees during the winter, they die inside the colony, the weather needs to be nice for the other bees to remove the dead bee from the colony, if I understand it correctly. But we have self-removal of the bees, that it's when the bee is infected by virus or with any other pathogen, she will fly out and get lost somewhere. This is what we know. But during the winter, if it's cold, no, she will not fly out.
Yeah, I think that's the message, right? If it's too cold, they cannot fly. They cannot remove the bees going to concentrate that inside the hive. So that might be an indication of something happening right there. Okay, Duncan Mason, thank you. If you can bring those questions from YouTube here, that would be great, man. That will help a lot. So because I can do this now, the question from, yes, so we have here. How effective would be OAV treatment during nectar flow by using 12 treatment regimen, treating every other day for 12 days treatment? It's a lot of labor. That's not my, with the commercial guys, if I come up with that idea, somebody's going to shoot me.
Maybe possible for hobbyists, hobby beekeepers, it's possible.
Yeah.
But every time, even with the oxalic acid, every time you are treating the colony, you are exposing the bees to oxalic acid, and maybe it's not good every day and every day. I mean, I assume I can't remember, I'm not in details. You need to just give them a little bit of time for the bees to recover again and then treat again. I'm not sure about that because I'm not in details of oxalic acid. Please just add to it, Humberto, because you are mostly working with beekeepers.
Yeah, I'm not sure the amount of treatment will be effective. You need to understand the cycle of Varroa. Majority of Varroa are inside the cell, so I think 80% is still inside, and oxalic acid might reach inside, but I think it's gonna be most effective in the adult bees and the bees that are exposed to the environment. So you need to understand where you are in the life cycle of that colony to do the amount of timing correctly with the OA to not only get rid of the first one from the adult bees, but and then have a specific timing where those that are hatching and new emergent bees to be exposed to the treatment as well. So I don't think every day. I think every week would be a good timing. So the new emergent that are gonna come every week, it's gonna be exposed. I think 3 times, 1 per week. I think you have a good regimen for your operation. But I think 12 will be. You might even hurt your bees. I'm not sure. So there is a lot of questions coming. So there is the gentleman, Duncan Mason, is doing a fantastic job helping me out here. He's putting the questions from YouTube here for us. So Barbara Keating is asking us, would you recommend shook swarming? I'm not sure I know what that is.
I think what they do is they remove the cap brood or brood combs, if I understand it correctly.
Is that to give an interruption in the?
To give an interruption for Varroa mite, yeah.
Oh, okay.
The understanding.
Yeah, this is one way to if you get rid of the brood, they don't reproduce, they die, and then you put the adult bees back, they build everything back again.
But then you need to have a good nectar flow. That's my understanding. Yeah.
Yes, a lot of resources. You need a lot of resources.
Because they need to come back quickly, or at least for a while. Because right here we install packages, and then we wait until sometimes, because older bees will die, the population goes down, and then recover again when the new generation comes. So you need to have a little bit longer time, plus have resources for building the colony.
All right. I hope I give you some information with that. Let's move on. Question from Anne. The four-moving virus is transmitted to workers chewing up pupa. Is there as virulent as when spread by Varroa? I think there is a recent publication
Exactly
from the Beltsville lab on that. I think so.
No, you are right, Humberto. It is with Eugene Ryabov, if pronounce his family name, and they very nicely show that when the bees are cannibalizing or removing the dead bees from the cells, because why it happens, why they are removing the bees, because that bee is infected or infested, they are sick, and that's why these bees that are cannibalizing or removing that bees, they become infected by the foreign virus. But about the virulence, what I can say is that it's just again one another way of horizontal transmission, that they digest the virus and virus needs to go to the digestive system, spread to the body, pass the digestive system to the rest of the body, spread, localize, spread, and make an infection.
Yeah,
And
the cycle continues. But one point that I think that might be interesting here, a lot of people try to push to select for bees that remove, and doing so, you're also selecting bees to get in contact more with those high infected viruses. And wouldn't that be counterintuitive? You're pushing them to get rid of them, and so therefore you touch them more, and maybe you get infected more.
yeah, that is another.
It is another angle to see the problem. You're thinking you are helping stimulating the behavior, but maybe stimulating the behavior, you are pushing them to get in contact with high infected bees. So I never thought about that. That was just an idea that just popped up in my head. So I don't even know what to think about. Okay, more questions for us. Here we go. What mite percentage would be low enough to ensure that drones and workers have low or no virus levels? Mite percentage. Ooh, interesting.
I think there are studies around that, but the thing is, we have at the beginning of the season we have low Varroa mite, and then specifically, what my understanding, I don't know if there is any study around that. I want just to connect it with the queen and queen production and the infection by queen, because we have this discussion right now. We are not talking about the general viruses things, but my understanding is that during the May, June, and a little bit of July, those queens, or this is my personal experience. Maybe I'm wrong, but this is my personal experience. Those queens that are produced around this time, because the population is in a high level, and high population normally are healthier than low population, the weak colonies. So these queens are emerge with low level of virus, and they are healthy and high quality queens. When we get to the end of the season, when the Varroa mite level is increasing and the virus level is increasing, so then the queens that are producing by then, they become infected after emerging. So that is what my personal experience says. Maybe I'm not right, as I said, but it seems that Varroa mite is a really, really game changer that we need to take care of. I mean, during the summertime when the Varroa mite, I mean, still the Varroa mite is there in the colony, but the population of Varroa mite inside the colony is low, and then that's
So,
okay.
yeah, so that question, yeah, I think I have an answer for you, Heather, but you might not like it. The number is zero. That's the only way to ensure that drones and workers are not going to have virus level and high enough to create problems. So try to keep your mites down as much as you can. And for some, there is many different philosophies regarding keeping bees. It's natural when you want to keep without treatments, and I understand all of that. But the data we have now, I think, to answer your question is when the mites are down, viruses are down. So controlling mites might be very good to control viruses. And yeah,
I mean, this is the answer that right now we have for viruses. Maybe
yeah,
future will change, but right now, this is the answer we
Right
have.
now, it's control mites. That's the best thing we can do to control viruses, is control mites. And I know some people out there don't like that. Organic farmers don't want to, I understand all of that. And we need to be creative and find another ways. But what the data is telling us now is that, at least what we have, we need to keep going, more studies, more technology. But the only way to help is to controlling mites. Another question for us. Are you ready?
Yeah, just go ahead.
Yes. Barbara Keating, would you encourage more drones by letting them drone more drone combs on large cells wax starter strips? So I think she wants to produce more drones. So production of drones, you're gonna have more drones, and maybe this is gonna dilute the amount of viruses on them. I don't know. I don't know if that's the question. Barbara, let me know if I'm going to the right direction. So I don't know if I would encourage more drones. Sometimes commercial beekeepers encourage drone production when we don't have them in the season and they need in specific time of the year to continue production of bees, for example. But Amiri,
Does she ask about the cell size?
yeah, if you put a
Because
if you put a specific size of cell there, you kind of stimulate the queen to produce drones. We know that. I think the question is if this induction in a different season where it's not naturally to have more drones, what will be the consequences to have more drones regarding virus infection levels? And I don't have an answer for you, Barbara. I don't know, to be honest. I don't know. But it's interesting. Thank you very much. I'll make a note. Another one. Will we ever see immunity or tolerance to viruses? Ooh, that's a good one.
well, what we can say right now, just reviewing the publications, I can say every time that people do survey on viruses, always there are around like 10% of the colonies that people survey, they could not find any or detect any virus in them. This is what we see it. And even I remember one of your publication, Humberto, you injected Australia QB paralysis virus to two different colonies, the pupae from two different colonies, and you found that there is a difference between the pupae from two different colonies. So it seems that bees have such that ability to control varroa mite, but we need to do more study for the antiviral mechanism to understand the honey antiviral mechanism, and understanding it will help us to have a shortcut to have bees controlling viruses by themselves. Yeah, of course they do have the mechanism to control them and immunity.
Jack, my take on that is this is for my personal experience in the lab. Just like Dr. Amiri said, I injected in the past thousands of pupa and larvae with viruses, and we have some specific genetic background. Some of these pupa and larvae do not develop the disease, even I injecting a lot. So I would say to you, yes, we have that. We might be able to select those very specific combination of the queen genetic and drone genetic that allows that larvae to survive the virus infection. However, in honeybee and everything in biology is always a combination of factors, and I'm pretty sure that specific background able to fight the virus disease might be missing something else for the normal biology of the bees. So that's why we're not selected in nature just yet. Maybe we can help with that, with some specific queen breeding programs to select more of that. I would like to see more of those programs. I know a couple laboratories and places that people are trying to do that. So I would say yes. I think we're going to that direction. But there is a lot of variables that we researchers. It's not like cows, and that everybody control everything, what they feed, how they mate. Bees are very complicated to do research. We cannot control what they eat. We cannot control how they mate, and that's where all these problems that we see happens all the time.
But what I can add here, Umberto, is I know there is a study going on in Baton Rouge, USDA Baton.
Being Baton Rouge. Mm-hmm.
Yes, they are selecting for a complete resistant to the form ring virus. But I don't know the progress. I don't know the result right now, and I cannot talk on behalf of them because I don't know anything. And that's why I'm just saying that there is a study is going on with that as well, just for people to know about it. That
Good to know.
Researchers are doing and working on. But the thing is, the thing is here with the system of, because you need to control everything in honeybees, and the honeybee in nature that we see it like a queen flies out to get mated with as many as possible drones from different genetic backgrounds. When it comes to selection and mating in this system, we are killing that and genetic variation that we have in the colony, because queen just flies out to get mated with as many drones from different genetic background, have this genetic variation inside the colony. This is the nature that is happening with honeybee colony. When we are selecting and mating, we are killing that variation in the colony. That is against that nature.
Which is very important. That flexibility of genetic background inside the colony allows the colony to be strong, because if something happen, just a small part of the population is going to be affected, and all other colonies are going to have genetic varieties of bees that will survive or be more tolerant for example, some virus infection. So when we kill that flexibility of variety in genetic background, we were also hurting the bees in nature. So it is a very tricky situation. If we got one side, we take the balance off. So quite complicated to control.
Exactly the same thing is happening with hygienic behavior, or some of the trait that people are selecting, and even they increase the hygienic behavior in the colony. And then the problem starts from there, because they need to take care of the genetic variation inside the colony. Plus, with the high level of hygienic behavior, more and more brood will be removed from the colony, and then the colony will not grow fast, or will not grow up after some level. That's another thing. So we need, for the honeybees, because they are different from other animals, and that's why it becomes more complicated. We need to take care of all these little steps and little part of the puzzle.
Yeah, but my hope, Jack, about that is that we start to understand better the immune system of honeybees. Even there is some technologies coming up with possible vaccination of bees. So we're still learning with that. And I believe that we don't know everything. So I'm pretty sure that soon we understand the immunity of honeybees in a social environment that they have. We're going to be able to make more tools or have more ideas or how we can help them. So I know lots of laboratories working on that, in this understanding of the immune system of honeybees, not only in the individual level, but in the social level, that as soon we get that level, that we have a full understanding, just like we have with the humans, that we're able to create all kinds of different vaccines, RNA vaccines, and all that kind of stuff. So more knowledge means more tools for us in the near future, and I'm always hopeful for that. Okay, Dr. Amiri, one more question. Dr. Malcolm Sanford, welcome back to the show. Dr. Sanford saying, what about testing bees for viruses, like testing humans for COVID? So if I understand correctly, Dr. Sanford, you want us to test every single bee on a planet. I like that, especially if I have a company that sell those things. I don't know exactly what you're trying to imply, Professor, so give me more information. So more questions. Here you go, Dr. Amiri, that's for you. So viruses get into the spermatheca. Are they also reproducing at high levels there? Or, so they even clean eggs would be infected? So that's interesting question. Were you able to demonstrate the viruses are replicating in those in sperms, or you just detect the virus there?
No, in the sperm we did detect the virus. I mean, it would be nice if we were like doing another technique and finding out if the virus was replicating in a sperm specifically you are talking about.
Let me just to clarify to people at home. So there is a big difference when you just detect the virus, and another way is you detect the virus, but you also demonstrate that the virus is infecting the cell, in this case, a sperm of the drones, because we could be counterintuitive. Okay, we detect it, replicate, might be damaging the sperm as well. So there is a lot of components, a lot of questions that come up with this idea of, okay, we have that in the spermatheca, but where, what cells are infected, are infecting, are replicating, or just passing through? So there's a lot of different scientific questions that we have there. And that's what I asked Dr. Amiri here. Have you ever not only detected, but demonstrate replication of the virus in any of the cells, the sperm or the walls of the spermatheca? Also, they have cells there. Have you ever been able to do some research on that to see how the virus are passing through this membrane and getting into the system of the honeybee queen?
Not really. We did not test it. It's just an open question. Even it is interesting, as you said, that understanding if the viruses are in the sperm, in the single sperm, and that was one of the questions that we had, and we answered part of it because we wanted to see when she is one infected queen is lays one infected queen lays egg. Uh, lays eggs, she is how the virus passed to the next generation. It is just inside the egg, outside of the egg, is coming by the sperm, or how this happening. And that's why we had queens. And I did not explain it explain it here in this presentation, but we found out that most of the deformed wing virus, when the queen lays egg, is outside of the egg and less inside the egg. I mean, inside the egg, we did not test it whether it comes from the sperm and how it comes, but there is, our study shows that still a little bit of virus, about, if we think about the size of the sperm and how many virus particles one sperm can pass to the egg, maybe it is reasonable to think that the virus can come with the sperm and enter the egg. This is possible to happen. Yeah, but no, we did not test the replication of the virus in
Yeah,
any.
because that would select a very virulent strain. Because in this case, if the sperm is infected, that means every single cell of the honeybee that come from that cell will be infected.
Yeah, one of the thing is that, and then the study that we have right now that is, I mean, it's close to complete. We would like to know, and that is in general. It's interesting because when you know when the virus is transmitting or how the virus is transmitting, you can find or develop a strategy to cut the roots. And the thing is the queen, one route that we did show with the mating. You saw it. You can just have the healthy drones, for example. And the other route is, so when the queen is laying eggs, when the queen lays eggs, if this mother queen is infected, so you can easily, and those infection can pass to the next generation and make the next generation infected badly. You can just replace that queen, and then have the next generation or new queens that you are producing from those eggs healthier, at least at some level. These kind of things, that is kind of a strategy to find an alternative to limit the viruses from very beginning of the life.
This is indeed a very fascinating subject. I know that was a overload of information. I just want to thank you everybody at home to stay with us for that almost 2 hours now, and we could keep going,
Yeah.
but I actually I have another appointment and I need to go. So I want to just stop this right here. Thank you, Dr. Amiri, very much for your time, for the donate your time to come here today and educate us all about how viruses infect honeybee queens and all this huge amount of information. And thank you for your work. Without people like you doing the hard work and doing this ingenious experimentations designed to get the data that we need, we'll never be able to progress to help the bees. And for that, I'm really appreciate your time here. Thank you very much. Thank you everybody at home to be with us. And I'll see you guys in the next video, InsideTheHive.tv, the show about bees. See you guys next week. Bye-bye.
Right.